The first speaker is Christy Rigby in Gothenburg, Sweden, who's going to talk to us about two of the favourites of Twilight Zone aficionados, copepods and diatoms. So over to you, Christy. Thank you.
Okay, let me just share my screen. And I hope that looks okay. Yes, good. Okay, hi everyone, I'm Christy, so I'm a PhD student in my final year at Gothenburg University, and I work in Erik Selander's lab, and today I'm going to talk to you about one of the projects in my PhD, and that's predator influences on diatom community size. So I work with a group of signaling compounds that are produced by copepods, and they're actually called copepodimides. These were identified in 2015 as a group of signaling compounds. And what copepods do is that they taint the seawater with these compounds, and then phytoplankton are actually able to recognize them and launch their defensive traits. So what we've seen before is that we get an increase in saxitoxin production, and we've seen an increase in domoic acid production. We've actually seen an induction of bioluminescence, so we've seen lyncodinium actually shine twice as bright with the copepodimides there. And we've also seen a reduction in speed in certain algae, and we've also seen that chain-forming diatoms are able to split up their chains. So I'm going to talk to you a little bit more today about the chain splitting and some results from that study. So just a bit of background, in case you're not familiar. Diatoms, they account for 20% of the world's primary production, and you get them either as solitary cells or in colonies. And there's several theories that exist around the idea of chain formation and the benefits. So some of the benefits of long chains is that they're favoured under nutrient-rich conditions, as the cells within the chains don't have to compete for nutrients. You also have a reduced sinking rate, but this is only for alive colonies. Dead colonies will sink faster than single cells. And you get a benefit of shorter chains as well. So here you'd get an increase in nutrient uptake, although in turbulent conditions, long chains actually outperform single cells. And the encounter rate is much smaller with zooplankton. It's much harder to find for a copepod to find you if you're smaller. So therefore it's sort of likely that nutrients weren't the driving factor for the evolution of chain formation. So here I look to see if these predatory cues, the copepodimides, played a role. So it's previously been shown that copepods can reduce the chain length in Skeletonema marinoi, and we know that the copepodimides are the cueing compounds, and we know that size-selective feeding is very common in the pelagic. So by reducing their chain length, these diatoms, it's thought to be a defensive mechanism, because if you become smaller, then the chances of you encountering a predator, in this case the copepod, becomes even smaller. So it becomes like a defensive mechanism. So I wanted to see if copepodimides can function as a general alarm signal and break up other chain-forming diatoms, not just the Skeletonema marinoi. So I picked 3 species from the university's algal bank. That was Thalassosira rotula, Chaetoceros cervicitus, and Chaetoceros affinis. So what I did was that I coat copepodimides, these compounds, onto the glass wall of a vial, and then I add the cultures. So here I had a starting cell density of 100 cells per mil at 5 replicates per treatment. So I had a control, a 1 nanomolar copepodimide concentration, and also a 5 nanomolar concentration. So I added the cultures, put them on the plankton wheel. This helps the cells stay suspended in the water. And then after 48 hours, I took down the experiment and measured the growth rate and the chain lengths. So what I saw, these are the results of the chain length. In the box plot here, each treatment has a total of 500 individual chain lengths, where 100 counts come from each replicate. And what we see here is that Thalassosira rotula decreased their average chain length by 79% in the 5 nanomolar treatment when compared to the control, and by 64% in the 1 nanomolar treatment. And what you can see on the right hand side here is that this is how it looks in the microscope. So here is the control, here is the 1 nanomolar, much shorter, and then in the 5 nanomolar we almost see no chains left at all. We saw the same pattern in Chaetoceros curvacetus. We saw a reduction in chain length by 49% in the 5 nanomolar and 28% in the 1 nanomolar. But for the Chaetoceros affinis, this one actually increased its chain length by 12% here in the 5 nanomolar and 42% in the 1 nanomolar. But when we did a GLMM, the results are not significant for that, but the rest are. So to expand this experiment a little bit further, I did a grazing experiment, and this was to confirm whether or not grazing pressure is actually reduced on shorter colonies. So I grew cultures, some with short chains, these were exposed to copepodomides, and some with long chains. And what I did was I divided them equally and then either added copepods or just had them as control. And then I had them on a plankton wheel, 20 hours, and I used a particle counter, and then I calculated the clearance rate. So the clearance rate here, that's the number of milliliters an individual copepod can clear per hour. So on the x-axis here, we have the equivalent spherical diameter, and this is used since chain-forming diatoms are not completely spherical. The solid line, black line here, this is the mean clearance rate from 4 replicates, and we have the standard error shaded in grey. And then the black line, this represents the neutral effect of copepods, so meaning no grazing. And then the green line, that is the distribution of particles from grazed cultures at the end. And also the blue one is the non-grazed cultures. So what you see here is with Thalassosira rotula, you point there, in the larger size classes you can see that it's swept clear here, whilst the smaller particles are not. So we can see that grazing is higher on longer chains here. For Chaetoceros curvacetus, we also see the same pattern. However, we do see a higher negative clearance rate here. I think that the copepods may be sort of attributing to these size classes by either not feeding on them or sort of stimulating growth, or perhaps sloppy feeding, like breaking up the longer chains into shorter chains. And for the affinis, Chaetoceros affinis, we didn't actually see any reduction in chain length from the copepodomide experiment. And what we see here is that it's not indicating any size selective feeding. So I'm thinking that they probably depend more on their spines for this species. So I wonder if they're sort of ramping up their spinal armor here instead. So yeah, just the main findings, that we see that copepodomides induce chain length shortening, and the grazing pressure is actually reduced on shorter colonies, so it's better to be small. And what does this mean in the terms of the ecological outlook? Well, the shortening is likely to affect the fate of the carbon, as sort of the vertical transport is likely to decrease, as the shorter chains will stay in the upper surface, and they're less likely to aggregate and form marine snow. And then you have the shorter chains, which also opens up for the microzooplankton to move in, thereby changing the levels of the trophic cascade there. And yeah, with that, I'd just like to thank the people that have helped me on this project. So I'm part of the Signals in the Sea group, and this is our website here. We can check out what we've been up to and our recent work, and then a couple of people that have helped me out during the project. And that's me.
Excellent. Thank you very much. Fantastic stuff. Thank you, Christy. Do we have questions? Let me just see if there's anything in the chat. Nothing yet, so people please do add things. If I can ask a question while people are thinking of their own. What is it that leads the copepods to exude these chemicals, given that it's clearly not in their best interest?
Yeah, so that's a good question, and we don't know why they're producing them, because it's not beneficial for them at all, but we have an idea that it's just a digestive sort of byproduct, because if a copepod is well fed, then they produce higher levels of these copepodamides. So I think it's something to do with their feeding, but yeah, we don't know what the benefit for them actually is at the moment.
And does I think Ken might have had the same? Yeah, I think Ken has asked the same question. And does it? I mean, you've done the work with, I guess, doing it in a laboratory. You have a restricted number of copepods. I mean, do you have any feel for whether the effect varies with the copepod that you look at?
Yeah, so there's actually some species that don't produce them, and they're like the ones that you find in rock pools and stuff like that. But yeah, different species produce different levels of the copepodamides, and specific compounds are, found in others but not in some. So it really sort of depends on the species as well. But
and there's no sort of relationship amongst the ones that do more or less in terms of, whether they tend to be open water species, deep water species, whether they feed on particular things. I guess I'm just thinking partly in terms of environment and partly whether, you are what you eat, whether what they're consuming is partly responsible for the release of the copepodamides.
I mean, we're just in Sweden, so we've only sort of tested the copepods out closest to us.
Sure.
But yeah, it would be really interesting. I'd love to collect copepods from sort of all over the world. We just got some from the Arctic region at the moment, and we're sort of testing what makeup of copepodamides they have. But yeah, it seems like for us that the colonists are like the mega producers of it.
That's interesting. You've probably come to the right place. We have quite a few devout copepod fans on this call, quite often. So it may well be that there are some people here who can provide you with a variety of copepods from different sources. So I'm using up all the questions. So has anybody else got any questions? Let me just check with the participants if anybody's got a hand raised. Does anybody else have a question for Christy? I guess the other one that I have in mind is in terms of you've identified the fact that if you move a colony from forming large chains to forming smaller ones, then you potentially impact the flux of material sinking out. Do you have plans to look at that in any way? I guess it's a little difficult in a laboratory.
Yeah, we did a little experiment with rolling tanks and aggregates.
Okay. Right.
We sort of did a pilot study, and we sort of need to continue with that one. But what we found was when you add the copepodamides with the rolling tank, they don't form like very nice compact aggregates anymore. They're very loose and very sort of fluffy. It's like they can't sort of, I guess it's doing something to the TEP production, that it's not sticky enough anymore. But yeah, that was just a little pilot that we've done so far.
Right.
But yeah, it's all sort of lab-based stuff. It's hard to get that out in a mesocosm in the field, really.
Yeah, but I mean, it does sort of indicate some of the processes, and the fact that the aggregates are forming in a slightly different way. And once again, that's very intriguing. So let me just do another check if anybody has any questions in the chat. elsewhere? I can't see any. As I say, please feel free to add any questions to the chat at any time over the rest of the call. Hopefully Christy can stay with us, and so if she sees any questions she can pick up. But otherwise, thank you very much, Christy. Really enjoyed your talk. Thank you.